have a complete strand of DNA. To replicate this DNA, a helicase enzyme comes and unwinds the DNA, leaving two parent strands. The single strand binding proteins come and prevent the parent strands from repairing themselves so that they will not bond together again and prevent the replication process. Next, a primase enzyme comes and begins the process. Because DNA can only build from the 3' end, but there is no 3' end to build off of, an RNA primer, an RNA chain called a primer, is created by a primase that starts the process. Now you have an available 3' end, and a DNA polymerase comes and helps bond these nucleotides. The DNA polymerase 3 removes the 2 phosphate groups, and this energy allows the bonding to begin. This means that you can create a chain. Now this is a leading strand because it's built in a continuous process because it can move in a 5 to 3 direction with the help of the DNA polymerase 3. The phosphates are released back to the environment, and the process continues to build a complete complementary strand. Now on the other side, this is called the lagging strand. Again, we have a primase come, which begins the process of the DNA replication by creating an RNA primer. Now the RNA primer begins to build something called an Okazaki fragment through the same process. Again, the DNA polymerase 3 comes, removes the 2 phosphates from the nucleotide. The energy released allows it to bond into a chain. And this chain is known as an Okazaki fragment. Again, we have a primase to start another Okazaki fragment. This lagging strand is the result of the 5 to 3, the fact that it can only build off of the 3' end. DNA polymerase again comes, removes the top 2 phosphates, allows it to bind. And the process continues. Now DNA polymerase 1 will take this RNA primer and replace it with the appropriate DNA. Replaces it with the appropriate DNA. So for example, here on the lagging strand, this is replaced with the DNA with the help of RNA polymerase 1. This is also replaced with the help, this one is replaced with the help of polymerase 1. And again, the top 2 phosphates are removed. However, these are 2 separate Okazaki fragments. What that means is that you need DNA ligase to bond the 2 Okazaki fragments together so that you have a complete strand. Now, the DNA polymerase 1 also acts on the leading strand. So here, again, the RNA nucleotide is replaced with a DNA nucleotide, completing the replicated fragment.